Hi everyone, welcome to my YouTube channel. So, as humans, what we see in our daily life is that usually a healthy human, has a pair of hands, has a pair of legs, a pair of eyes, and other morphological features. But uh, at a deeper level, it is not that simple because we humans and any living thing on this earth is a complex structure of small atoms, or say it is a mixture, it is a living mixture, it is a working biological system of carbohydrates, proteins, nucleic acids, amino acids, vitamins, minerals, etc. So nearly all the elements that are present on this earth's crust is also present in our body. So it is very surprising that we are the same, we are made up of the same elements that made up this whole universe, but it is what it is. So we are going to look up about the same compounds, or say the same structures, the complex structures, but we will start building it through base. Okay, so this series is about decoding biochemistry, and we are attacking, or let's say building the foundation, that is the biomolecules. Okay, if we understand biomolecules properly, then we will never face any difficulty in understanding any concept of biochemistry. Okay, because this is where the base, where the foundation lies. So without any further ado, let's start with the lecture. Let's assume that this is a living tissue. Okay, let's say this is a tissue of a leaf. Now we are going to analyze something about this tissue, and we call this process as tissue analysis. Now this living tissue, okay, it can be anything. I've taken an example of leaf. Okay, it can be anything. It can be a tissue of a rat. It can be any organ also of any living organism. It can be the liver of chickens. It can be the earthworms also, for that reason. Okay, it can be anything you can imagine about living organisms. Now this tissue is grinded with trichloroacetic acid, whose molecular formula is Cl3, that is 3 atoms of chlorine combining with the acetic acid. So therefore it is called as trichloroacetic acid. So we grind this tissue in a trichloroacetic acid in a mortar and pestle. Okay, mortar and pestle is nothing but this kind of thing which we use in our kitchens. So in laboratories also we use it for not very, in laboratories mainly we use it for an experiment which does not require much sterile or aseptic conditions. Okay, so when we grind this tissue with the trichloroacetic acid, we will get a thick slurry. Okay, in which there is lysis of all the cells that are present in this tissue. So these cells will lyse, they will break apart. Now this slurry is subjected to sieving in the sense like how we sieve the daily chai of like our evenings also. We use a sieve to sieve the flour. We use a sieve to separate the tea leaf from tea. So similarly we are going to use this system where there is a beaker, okay, a laboratory apparatus called beaker, and we are going to overlay a muslin cloth on its upper region. So we are going to fix this over here such that the muslin cloth covers the opening of the beaker. Okay, now this muslin cloth will act as a filter, and whatever this slurry is there, we are going to pour it over the muslin cloth. Okay, so what happens is that anything that can pass through the holes of muslin cloth has very minute holes in it. Okay, so anything that can pass mass will drop down into this region of the in this container of the system, and all the denser molecules will remain above. Okay, there might be some pieces of tissue, there might be something uh, like heavy molecules and all, which will remain over here. Even let's say chlorophyll also, for that matter, if we are assuming leaf, then the green pigments and all may remain. Okay, so whatever remains above the muslin cloth is called as retentate, because it is retained over the muslin cloth. And whatever comes down through the holes of muslin cloth, we call it filtrate. Filtrate is also written as like this also. Both the spellings are correct. So whatever we get in this container is the filtrate. Now we are going to distinguish between the retentate and the filtrate. Now filtrate is acid soluble. Okay, whatever contents are there inside this beaker, they are acid soluble, because see, the tissue and the components of the tissue were soluble in this trichloroacetic acid. That is why they have become like a team. Okay, these two components, the components of tissue and the trichloroacetic acid, have become one substance, and they have got down through the holes of the muslin cloth. So therefore we call it acid soluble. And the retentate is acid insoluble. That is why it is remaining above the muslin cloth. It has not sieved through the muslin cloth. Next we are talking about the size of the molecules. Okay, these words micromolecules and macromolecules are important. Micro means they are very tiny in size. Okay, macro means they are comparatively larger in size. Okay, micromolecules which are present in the filtrate are usually monomers. We are going to look forward about these terms in our next lectures. And so for now you understand that filtrate will have usually monomers and retentate will usually have polymers. Okay, we are going to understand why we will get something like this, why it is not the other way around. Okay, next we'll also see some examples. Okay, what can we find over here? Okay, so it can be nucleotides. Okay, it can be amino acids. We will study these components in detail in the further lectures, but for now you understand that there will be something called as nucleotides and there will be something called as amino acids inside the beaker. Okay, and on the muslin cloth what we can expect is proteins and lipids. Now see, lipids are not macromolecules. Okay, this is an exception over here. Lipids are not macro in size, they are micro. Okay, lipids, the structure of lipids is something like this. It has a head and it has a tail. Okay, now this tail is afraid of water. Okay, in science we call it as hydrophobic, and this head is hydrophilic, means it loves anything that is aqueous, it loves water. So there are many this kind of structures present in a cell. Okay, it will form lipid bilayer. It is present in the plasma membrane. So usually what happens is that in such kind of experiments, these lipids will form aggregates. Okay, they form micelles. It can be double structure also, like inside also there can be some head regions also. Like imagine like this. This is a lipid bilayer. Okay, so in similar structure only they'll form an aggregate, that is they'll form a circle like this. Okay, and that is why they will remain over the top of the muslin cloth. Okay, so this was the video about tissue analysis. In the next video we are going to look about ash analysis. Okay, I would recommend you to watch this first, understand it properly. If you are not understanding something, you can drop down it in the comments. You can drop down your doubts in the comments. I'll try to answer them in the next video at least. So this was about tissue analysis. The important terms over here are trichloroacetic acid, and these terms retentate, filtrate, and their properties: retentate, filtrate, and retentate. Yeah, one thing I missed about filtrate is that there can be many inorganic elements also. Inorganic elements in the sense calcium, magnesium, all these kinds of elements which are there on the periodic table. Okay, so they can be also present in the filtrate. But in retentate, 99% of compounds or molecules which will be found on the top of the muslin cloth, they will be carbon based. Okay, hardly there will be one or two impurities which will be having inorganic elements. So that is it for tissue analysis. We will look after ash analysis. It is also very much interesting. It is very simple. So to summarize about tissue analysis, understand that a tissue is taken. It can be any living tissue. It is grinded with trichloroacetic acid, which will form a thick slurry, and after sieving we will get something called as retentate, and we will get something called as filtrate. Now looking up after the definitions or distinguishing properties of filtrate and retentate, remember about solubility, the parameter solubility. Filtrate will be acid soluble, whereas retentate will be acid insoluble. Filtrate will contain micromolecules, retentate will contain macromolecules. Filtrate are usually monomers. Filtrate will usually have monomers, that is, these micromolecules which we are expecting will be monomers only. But here they can be polymers also. Okay. Examples are nucleotides. Examples, the other example is amino acids, and here the examples are proteins, which are polymers of amino acids. And there is also one exception which is also asked in many entrance exams like NEET exams, NEET PG, NEET UG, and also some other exams like NET, SET, etc. So that was it for tissue analysis. Just remember the words and it is very easy to understand. So lipids, I was talking about lipids, I forgot. Lipids will be forming aggregates. That is why they will remain on the top layer of the muslin cloth. Okay, they won't be passing through the muslin cloth. So that was it for this video. Thank you.